Spermatogenesis occurs in males, and it begins with a diploid spermatogonium in the seminiferous tubules, which divides mitotically to produce two diploid primary spermatocytes. The primary spermatocyte then undergoes meiosis I to produce two haploid secondary spermatocytes. Oogenesis starts with the process of developing oogonia by the transformation of primordial follices into primary oocytes. This occurs in females. It is complete either before or after birth in humans. Meiosis I and Meiosis II for both spermatogenesis and oogenesis are similar for except a few things, which is probably the ending. In Meiosis I, during the prophase I, chromosomes and individual chromosomes, nucleolus and nuclear envelope disappear. Homologous chromosomes come together as pairs by synapses, forming a tetrad. Centriole move to opposite poles with spindle fibers between them. The process of crossing over happens here in prophase I. Metaphase I is when the homologous pairs of chromosomes align along the equator of the cell. The two chromosomes attach to one spindle fiber by means of kinetochore of the centromere. Anaphase I is when the spindle fibers contract homologous chromosomes, separate and move to opposite poles. Sister chromatids remain attached at their centrosomes. Nuclear envelopes reappear, spindle fibers disappear, cytokinesis divides cell into two during the final phase, which is the telophase I. In meiosis II, nuclear envelope and nucleolus, if reformed, disappears again. Centrally move to opposite poles, forming spindle fibers between them. Metaphase II is when the individual duplicated chromosomes align on the equator. One chromosome per spindle fiber attached by means of kinetochore of centromere. Centriole has back the poles. Anaphase II is when the spindle fibers contract and the duplicated chromosomes split in half. During telophase II, daughter chromosomes have reached the pole. Two cells invaginate and form four daughter haploid cells. They uncoil and form chromatin. Nuclear envelope and nucleolus for around chromatin again. Centriole for centrosome. The difference here is that spermatogenesis produces sperm, while the other one, the oogenesis, forms eggs. And both of those are necessary for the creation or the process of making human life. Gametogenesis is the process in which the creation of life is being ensured. The production of male and female gametes have to undergo complex processes in order to be molded for the role in the creation of life. In philosophy, in order for man to be fully human in the Greek ideal of full development, as said by Michael Moga, you have to develop your different potentialities. Man has to ensure that his physical, mental, communication, and social skills are fully developed in order to be a fully developed human. It is a complicated process as man goes through different challenges just for him to attain his role. Upon developing, though, he can now be truly human by using it to help better the state of the society. Just like in gametogenesis, our cells have to go through a complicated process in order to fully develop. Each of us differs in roles, just like the products of spermatogenesis and oogenesis. Both the sperm and egg are necessary for the creation of life, and whatever the product is during the development period in the Greek ideal of full development, those are all necessary for the betterment of our society. So that's all about these gametogenesis, spermatogenesis, and oogenesis understood in a philosophical lens. Thank you.